Hello friends, I am Ayantika and in this video I will be discussing about an overview of pteridophytes. The video will include topics of fossil pteridophyte, both the arborescent as well as the very minute pteridophytes, their distribution and their and extinction period in the geographical time scale, and about the living, that is the extant pteridophytes, their distribution, their general character, their reproduction, as well as the stellar organization. And I will conclude the video with the ecological and economic importance of pteridophytes. So this will be an holistic overview about various topics of pteridophytes. So let's start the video. The bryophytes are the amphibian of the plant kingdom, which means that they were the first plant to have both amphibious habitat, the water as well as land habitat. They were the first plant to inhabit inland, but they did not have a well developed vascular system. In fact, they didn't have a vascular system, so which limited their distribution, as well as water became a limiting factor. Due to their lack of vascular system, most of the bryophytes, they were ephemeral, they were not perennial, and also they required a huge amount of water to conduct their life cycle, especially their reproduction. So due to the absence of water conducting system, bryophytes were limited only during the wet season of the year and in the wet environment, that is by the side of lake and by the side of any other water body or vernal pools or temporary water bodies. So pteridophyte was a much better adaptation, and the pteridophyte, they formed the intermediate between bryophyte and gymnosperm. Pteridophyte are the first plants to have the vascular bundles, the xylem and the phloem, which helped in water and food conduction. So armed with the vascular bundle, pteridophyte had a much better adaptation, and they were not ephemeral, they are perennial and sometimes annual. And due to this conducting system, they were able to survive for long time, and also they were had a better geographical distribution, much far wider geographical distribution than the bryophytes. But eventually, the pteridophyte lost the battle to the gymnosperm, and most of the pteridophytes, which were very common during the Carboniferous period, were lost in the Permian Triassic extinction. We know that there are five mass extinctions, that is the Ordovician extinction, the Devonian, the Permian, the Triassic, and the Cretaceous extinction. So the pteridophytes, mostly the arborescent pteridophytes that were common during the Carboniferous period, so they were lost during the Permian extinction to the gymnosperms, the conifers, which had better adaptation for water, to deal with the water storage. So and water conduction. So we get a lot of fossils of fossil arborescent pteridophyte, as well as fossils of pteridophytes, very small pteridophytes in the Devonian rock layer, the Silurian layers. And in this picture we see three fossils. The first belonged this one belonged to Sigillaria. The second one is the Lepidodendron. This one is the Lepidodendron, and this one is the Calamites. From the cast, the fossil cast we can easily see that these are casts of stems, and as we see the region furrow and the and in the in the furrow, in between the two furrow, we see that the traces of the scales of the, not scales but markings of the leaf area is seen. And in lepidodendron also we see the markings of the leaves where once leaf was there. And this calamites has a very jointed appendage like stem. Nevertheless, the sigillaria, the lepidodendron, and the calamites, all they were arborescent tree lycopods. They were related to the lycopods. These pteridophytes were huge in their size, that is from 80 feet to 160 feet. The sigillaria, lepidodendron, calamites are three, but there are many other pteridophytes which were arborescent. They had huge trunks, a large diameter, and they reach great height, and their crown, the crown area had a very cone shaped, and they have a top crown that is cone shaped. And this in the top area they were dichotomously branched, or this dichotomy were again dichotomous, and this formed a cone shaped crown that were common in most of the arborescent lycopods, arborescent pteridophytes. So this sigillaria, they were found mostly in the sigillaria, lepidodendron, and calamites. They were mostly found in the subtropical and tropical region. They were found and they were not found close to the pole, but in the intermediate region, that is tropical, subtropical, both in the continent of America and Europe. They are very much found. And this sigillaria, lepidodendron, calamites, their fossil cast has been found in mostly this sigillaria was found very across America, also Britain, and many other countries like Korea, Tasmania. So this lepidodendron, there its fossil was found in only
Here we see
North
this
and
is
South
the
America.
pteridophyte.
The
This is
calamites
the
were found
picture of rhenia.
this is
Many
the
places
fossil
in America.
of, so these are the arborescent
Horneophyton,
pteridophytes.
and
There are also
this
very
belonged to the
small pteridophytes like
Xylon,
and this belonged to Cooksonia. So we see that these the size of this fossil, they are very small, few centimeters in height, and they, all these fossils, they were found in the mostly in Scotland, and some fossils were also found in America and Australia. But the small pteridophyte fossils, they were mostly found in Scotland, Aberdeenshire, that is the Rhine chart bed. And these are the very small fossils we see, and we see that the stem, it is dichotomously branched and repeatedly branched, and they have got the reproductive body, the sporangia, here at their terminal portion. Earlier in the arborescent pteridophytes also, they also contained terminal sporangia, but the small pteridophyte, they have a homospora sporangia, whereas the arborescent pteridophyte that I have shown in the last slide, they had a heterosporous sporangia, that is they had microspores and macrospore and the megaspores. So these are
in
the
geographical
fossils,
time scale,
these are some of the fossils.
we see
There are
that,
many other fossils, but if
Carboniferous period,
I'll make a separate
the-
video
this
on the
period
fossil pteridophytes,
these are
showed
just
the
few,
maximum abundance of the
pteridophytes.
and they were lost. They were lost during the Permian extinction. So during this period, Carboniferous period, it were abundant, and in the Permian period they got extinct. They got extinct in the Permian period, and They were very abundant during the Carboniferous period. However, we can find fossil evidence in the during the Carboniferous period rock. Also, we can find especially Rhynia, Horneophyte, and Cooksonia. All these we find the fossil evidence in the Rhine chart in Aberdeenshire. They were all mostly Devonian and Silurian. So here we see that this pteridophytes, they existed even when the dinosaurs, they even before the dinosaurs roamed in the earth. So we see it's had got a huge historical evolutionary importance, as because they were very much primitive plants, and they had got so much adaptations for land habitats, and they were in fact the first plant to have the vascular system, which later helped in evolution. So it is of tremendous evolutionary importance, especially their stellar organization, and I'll be discussing the stellar organization in the later slide. So this is how the arborescent pteridophytes they looked, and notice that they have attained great height, that is some few feet to even 100, more than 100 feet. So this lepidodendron fossil, which is very common, I discussed in the first slide. And also did I discuss the sigillaria, that is the first picture of the first slide was sigillaria. The second picture was the lepidendron, second picture of the first slide. And then there was also calamites, not in the diagram. Here we see that they have got a very long stem, and we see that repeated dichotomy, and they have got a semicircular hyperbole. Some have got a conical hyperbole like that of the conifers. So whatever be the shape of hyperbole, they are very much, they have got terminal strobilus, and the stem, stem, they had they were not very smooth, but they had got either ridges and grooves, or they have got leaf scars on the entire stems. The leaves were either spirally arranged, or they were arranged in a hole at the nodal region, or they were arranged decussately. But whatever be their leaf arrangement, they had a protostelic vascular bundle, and they had heterosporous strobilus at the terminal portion of their branches, and the heteropolous thallus, they have the microsporous thallus were many in number compared to the megaspore. Megaspore were few, and this even the fossil of the megasporangium and the microsporangium has been found of these fossils. So all these and about the distribution, they were very common. They were mostly found in this region, of America, in Canada, also some regions, and in Scotland, in some African countries, and even in the Southeast Asian countries. And this region they have been found, and in India they have been found in the Himalayan region, and certain parts of Madhya Pradesh, and along the Western Ghats, also some, especially Panchmari. Many of the species of Equisetum, Selaginella, Lycopodium had been found in this region of India. And apart from that, this is the global distribution of fossil pteridophytes. However, arborescent for pteridophytes were found mostly related to in the Canada, in America, and very small pteridophytes were found in Scotland and this East and Southeast Asia and India. They had very small pteridophytes, that is the Rhynia, not Rhynia, Lycopodium fossils, Equisetum. So these fossils were found there. Classification and also of the Himalayan region, that is the living pteridophyte. According to the Pteridophyte phylogeny group 2016, the pteridophytes, they had been divided into two classes, Lycopodiopsida and Polypodiopsida. So Lycopodiopsida contained Lycopodium, Selaginella and Isoetes. So apart from that, it also contained many other plants, but the Lycopodiopsida, these are the most important. They are actually covered in most of the MSc and BSc syllabus, so I mentioned this. And there is another class called Polypodiopsida. So Polypodiopsida, it is a wide class. It includes ferns like Teres and Salvinia, and it also contained Equisetum, Xylotum, Ophioglossum like plants. The Xylotum, this one is Xylotum. So we see that the modern classification has two classes, Lycopodiopsida, which contains only fewer family, but this Polypodiopsida, the second class, it contained many families, and it is a much wider class, and it includes both Equisetum as well as the ferns. So this is a recent classification, 2016. So while doing classification, studying classification, make sure what type of classification your university follows, whether they are still following the older classical classification or this new one. If you want me to make a detailed video on the new one, I write it in the comment section. I'll make a separate video on this classification. Now, this pteridophytes, they are the first vascular plants. That is, they had the first vascular system. But still, there is some difference between the pteridophyte and the angiosperm. Because the pteridophytes, they do not have have flower or seed, but they instead have a sporular method of propagation. They have spores, and this pteridophyte, they had 13,000 species, and they are mostly, they prefer cool and shady places, and temporary moisture is also required, because moisture is required firstly because it helps in the growth, but also one thing is that the reproductive male reproductive gamete, that is a sperm in the pteridophytes, they are either bi or multiflagellated, which means that for fertilization to happen temporarily, there should be a certain amount of water, especially the sperm, the sporangium containing the sperm are born in a higher position than the macrospore producing strobilus, which are placed in the lower position. So water is very much essential for the sperm to swim back to the egg region. So that is one thing, that is they require moisture for their reproduction, and apart from that they are mostly terrestrial, but however some aquatic species are also found. Some species are found to be growing epiphytically on other tree. So here we see that these are the terrestrial ferns, and this is the epiphytic fern that is growing on another tree branch, and this is Salvinia. This one is Salvinia, which is an aquatic pteridophyte. And mostly the apart from Salvinia, the other ferns, they usually require a very moist environment, and they are very much found in the tropical as well as the temperate region, but mostly in the tropical region we have a great variety of ferns. And their size varies. They can be very small, like Lycopodium selaginella, to very large tree like, for example, this Cibotium and Equisetum. This is an example. This is this tree like plant which might appear a palm tree like, but it is not palm tree, it is a fern that goes by the name of Cibotium. The scientific name is Cibotium. And here you see, this is the Equisetum giganteum. This is the Equisetum giganteum. You see, an adult person is standing, and this Equisetum is much, it is double the size of an adult human being. So you see that there is great variation in the size of the pteridophytes. Also, there is great variation in the in the habit. Like some of the pteridophytes, they grow erect. For example, this Equisetum and the ferns. Here, this Equisetum, and certain other pteridophyte, they are found to be growing. They have a creeping rhizome that branches and rebranches and gives off aerial branches. So this type is found in Lycopodium. Also, certain Lycopodium and as well as Mesipteris, they are also pendulant in nature. This is the Lycopodium that is showing this pendulant habit. And so we see that there is also great variation in their growth habit, as well as their size, as well as their habitat. So this is the overview. And this entire this subtropical as well as the tropical region, this we find many of the pteridophytes growing in this entire region. In the tropical region, in this tropical region, we see great abundance of the ferns. In the in the tropical region, we find great abundance of the fern, and in the subtropical region, we find great abundance of other type of pteridophytes. So, in this region, we have got huge amount of ferns, and in this region, we have other type of pteridophytes. And in India, pteridophytes are mostly found along the Western Ghats, more and much more along the Eastern Ghats. Also in the Panchmari region, that is the Satpura region. Also in the Himalayan region. So in this region, we have great abundance of pteridophytes and ferns.
Pteridophytes are the
Pteridophyte
first
and
plant to
fern
have vascular
and,
bundle.
and in the vascular bundle, they had tracheids in the xylem and sieve cells and the sieve parenchyma in phloem. And the type of stele is protostele in most of the pteridophytes, except the ferns, which has either siphonostele, eustele, or atactostele, or any other stele in between. And this is because of the fact that fern at immature condition, they are protostele. But later on, as the fern plant attains maturity, then it is the protostele is replaced by siphono or eustele or atactostele. This is because of the fact that in other type of pteridophytes, for example Lycopodium, Selaginella, Xylotum, the leaf trace does not have any leaf cap. But in ferns, we see that leaf trace has got leaf gaps, and due to this presence of leaf gaps, the stele appears stele appears much divided. So the pteridophytes, they have two generations. In the alternation of generation, they have a diploid sporophytic generation and the haploid gametophytic generation. And the sporophytic generation, which is diploid, it is dominant. And in this respect, that is, the pteridophyte have sporophytic or diploid generation as dominant, it differs from the bryophyte. It differs from bryophyte in many facts, in many features. In fact, the bryophytes do not have a vascular system, unlike the pteridophytes. The bryophytes, they have a dominant phase, that is Riccia, Markensia, this type of bryophytes, they have dominant phase as a gametophyte. If you notice, the Riccia, Markensia, or even the Sphagnum, they have got reproductive organs. In the dominant phase, they have reproductive organs, and the sporophyte, that is the food seta capsule type of that is a sporophytic generation, it is not only born in the gametophytic dominant phase, it is born inside the gametophytic phase, but also it is nutritionally dependent. The gametophyte of the bryophytes, they supply food to the sporophyte. So in bryophytes, the sporophyte or the diploid generation is dependent upon the gametophyte or the haploid generation, and the haploid generation is dominant in the bryophytes. But here in pteridophytes, our topic pteridophytes, we see that the dominant generation, it is the diploid or the sporophytic phase. Here in this figure, we see a sporophytic plant, where sporophytic pteridophyte, where they have a creeping, this portion, the creeping rhizomatous creeping portion of stem, and this creeping portion by dichotomy later on gives to aerial type of stem, this one. And they have also got root, and the leaves, they are spirally, here in this picture it is spirally arranged around that of the stem. So the entire plant body is divided into root, shoot, and stem. And on the tip of the stem, that is terminally, the strobilus is born. The strobilus are the spore bearing unit. The sporophyte or diploid generation produces strobilus that contains spores, which are haploid. So the entire plant is diploid, but the strobilus, which contains the spore in the strobilus is haploid. That is, meiosis is occurring inside the spore mother cell in the strobilus. So this portion, which contain the spore region, the only the spore is haploid, and the spore, it falls to any suitable ground, that is especially the moist ground, and it forms this gametophyte, which is also haploid. And inside the gametophyte is produced the sex organs. And here in this picture, we see a young sporophyte, that is diploid generation. It starts to grow from the gametophyte. So two generations. The gametophyte, here in this picture, it is growing above the ground, but nevertheless there are also certain gametophyte which are subterranean, that is occur below the ground, and such type of gametophyte, they are colorless or pale or brown colored, and they are saprophytic in nature, that is they are dependent upon diluting organic matter by extracellular enzymes. So they are saprophytic, and those that live above the ground, they are green and they are photosynthetic. So this is the gametophyte in this picture, and this one is the sporophyte, and this one is the gametophyte. Now we come to the sporophyte. We deal with the sporophyte first. So we see in this picture they have a subterranean rhizome, and we see the roots. So here we see the roots, but in certain species, especially Xylotum and Mesipteris, entire rhizome like stem, that is a creeping stem, entire stem has equally distributed rhizoids. So these are the rhizoids. These are the rhizoids, the filamentous rhizoids. These are the filamentous rhizoids, and here we see what is this? These are not rhizoids but root. So in pteridophyte we see two type of absorbing organ, that is the rhizoids and roots. And rhizoids are found in Xylopteris, Xylotum and Mesipteris, but roots are found in other type of pteridophytes. And the creeping rhizome, they are dichotomously branched. This creeping rhizome, this creeping stem, we see that there is dichotomy. Here there is dichotomy. This is the aerial portion. This one is the aerial portion. And we see that it is in dichotomously branched. Leaves of the pteridophyte, they have three type of phyllotaxy, that is a leaf arrangement. First one and most common is the spiral arrangement that we find in the ferns. And the spiral arrangement, here the spiral arrangement is found, and also decussate, that is this type of arrangement is also found, and that is found mostly in Lycopodium, for example Lycopodium alpinum. Also they have a whole phyllotaxy, for example in the equisetum they have, this is the stem of equisetum, so they have a whole of leaves, sheathing leaves. The leaves sheath the stem, so they have also got a whole type of phyllotaxy. So three type of phyllotaxies are found, and leaves are also two types of leaves, that is a microphylous and the megaphylous. The microphylous leaves, that is where the lamina has only one mid vein, and the macrophylous leaves are much larger in size, and the leaf vein has got many ramification. So there one leaf vein, it is getting ramified. So there is difference in size between the microphylous and the megaphylous leaves, and also there is difference in the vasculature. So vasculature of the megaphylous is reticulate, and microphylous, they have only one mid vein and no ramification. So these are the 2 points that differ, that by which the microphylous leaves differ from the megaphylous leaves: size and vasculature. So the leaves are usually sessile, that is they do not have any pedicel, and they can be ligulate or illigulate. Usually if this is the leaf, so there is a tongue like structure that is called the ligule, and if that is absent, it is called e-ligulate. And leaves can also be based on the transverse section. Leaves can be also isophilous, that is both the sides of the leaves are of similar appearance, there is not much internal differentiation. Or leaves can also be anisophilous, that is the ventral and the dorsal surface, they are two different. So anisophilous leaves are also found, and especially the anisophilous leaves are found especially in the ferns and all. And also one thing is that the leaves, they sometimes they bear the the sporangia on their upper surface or lower surface. And the vasculature of the stem, it is mostly protostelic. And but except the ferns, which is siphonostelic or actinostelic or eustelic. Then we come to the root. The root, it also has got a protostelic organization, and the roots, they appear endogenously, which means that say for example, this is the stele and this is the pericycle, and this is the endodermis that I am drawing, and this is the, for example, this is the cortex, and mind my bad drawing, but this is the epidermis. So if any plant, we say they have a endogenous root, then that root is derived from the endodermis. They have a root that originate from the endodermis or the pericycle. They are called endogenously developed root. So root in pteridophyte, they are endogenously developed, and they develop from the prostate, from the lower side of prostate stem, or they are developed from the basal part of the erect stem. So we see that they develop from the lower side or the prostate part of the stem. So they are adventitious there, because in all the pteridophyte, the primary root, they are soon replaced by the adventitious root. The pteridophytes, they do not have a seed or flower, so they reproduce by the spores, and the spores are the haploid structure, or they are the haploid structures that germinate to produce the gametophyte or the haploid generation. And the spore, they are born on the sporophytic or the diploid plant, and they are born on leaf, and that the leaf that bears the spore, they are called the sporophyll. But they can be present, if this is the leaf, they can be present on the upper surface in the axial position, or on the lower surface. Say for example, they are born on the lower surface. For example, here in the fern, we see these red dots on the back of the fern leaf, and these small red dot on the ventral surface, they are the sorus. That is, many sporangia occurring together, they form a sorus, and they have a covering, protective covering, and that protective covering is called the indusium. So indusium and containing the sorus, all these are present on the lower surface of the fern. And here in the Lycopodium, we see that the sorus, the sporangia, it is born on the upper surface at the axial position, and all these sporophylls, that is the leaf containing the sporangium, they are very closely arranged, very tightly arranged to form this strobilus like structure. So we see that the spore, they are both born on both microphyll as well as the megaphyll, that is the large leaf as well as a small leaf. But what will happen in aquatic species? The aquatic species, they have sporangia inside this sporocarp. This sac-like structure, they are called the sporocarp, and inside this they contain the sporangium. So this is this is found in aquatic species of water fern like Azolla, Marsilia, Salvinia. So these type of aquatic species, they have sporocarp. And the development is of the spore, it is eusporangiate or leptosporangiate. So leptosporangiate species are those species where the spore is formed from a single initial cell, and such type of sporangia are usually small in size. On the other hand, this eusporangiate species, they are produced from many initials, and as a result, they are much massive in size. And the eusporangiate species are Lycopodium equisetum and the fern Maratia. And leptosporangiate species are this Pteridium, Dixonia, and Cyathea, and many other ferns. So, the spore development is two types: eusporangiate and leptosporangiate. So, the sporangium in structurally, here we see the xylotum sporangium. Structurally, it consists of many layers, and of the many layers of the many layers of the cells that that form the sporangium, the innermost layer, say for example, there are many layers. The innermost layer is the tapetum. The innermost layer is tapetum. The outer layers, these layers, they have much thick wall and they help in protection. So the outer layers are protection, and the innermost layer of the outer layers, they are nutritive. Tapetum, usually the cell, they degenerate after a certain period and serves a nutritive function for the developing spores. Now the spore mother cell, that it undergoes meiosis or reduction division to form 4 haploid spores. Each spore mother cell produces 4 haploid spores, and the spores, they are disseminated by wind, and they fall on suitable substratum to form the gametophyte. So usually the sporophylls, they are either microsporophyll or they are megasporophyll, that is the small sized sporophyll or the large size sporophyll. But in equisetum, they do not have a leaf like sporophyll, but instead they have a very peltate or shield shaped sporophyll. So this was what I was talking. They are born at the here at the lower end of the disc, and the sporangia, they are contained on the lower surface of this sporangiophore. So the sporangiophore here, it is not leaf like, and here we see this was what I was talking about, and here this is in isoetes. The leaf on the adaxial or the upper surface, it contains sporangia, and this is also a sporophyll which contain the sporangia on the adaxial or the upper surface. This is the strobilus of equisetum, and this is the strobilus of Ophioglossum. We see that they have a compact sporophylls arranged together to form the strobili. This is how the fern, the sorus, they are born together. And here, this is the structure of a spore. We see that the spore, they have elaters together, that is joined on the uppermost layer of the protective layer of the spore. They form the elater and help in the dispersal of the spore. From the shape of the elater, we can easily see that the spore, they are dispersed by wind current. So the sporangia, they are two type. One is the homosporous, where the size of the spore is equal. Another is the heterosporous. In the heterosporous species, they have two type of sporangia. One that contains smaller spore, that is the microspore, and another is the megaspore. Here in this picture, we see that this is the microsporophyll containing the microsporangia, and these are the microspores. And here the megasporangia containing the megaspore. The microspore, it produces gametophyte that usually it generates the antheridium or the male sex organ. And the megaspore here, this one, when it germinates, it produces a gametophyte that produces the female reproductive organ or the archegonium. So these two type of spores in the heterospora species, two type of spores produces two type of gametophyte. But there are also certain species where a single strobilus, here we see here in this picture, in this picture we see that a single strobilus contain both microsporangium. Here this one is the microsporangium and this one is the megasporangium. So we see that these two species, these two type of strobilus, they contain both the megasporangium and the microsporangium. So there can also be strobilus that contain entirely microspores and entirely megaspores, but mixed type of sporangia are also found. But in equisetum, most of the species of equisetum, they have incipient heterospory. Incipient heterospory is found only in equisetum, where in the development of the spore, whether it will develop into mega or microspore, that will depend upon the fertilization criteria. If already there is a little chance of fertilization, then the number of megasporangia will increase. So this is the incipient heterospory. And the two type of spores, that is a homosporous and heterosporous
So
species,
the gametophyte
with the two type of sporangia. The spore, they produces gametophyte. The male, the microsporangia produces male gametangia, and the megaspore produces female gametangia. But when it is a homospora species, then what will happen? When in case of a homospora species, the gametangia will contain both antheridia and the archegonia. For example, in this picture, we see that the antheridia is also present. At the same time, archegonia is also present in the homospora species. But the gametophyte or the prothallus that contains of the homospora species, it will start the development of the opposite sex organ at two different time. That is, even if there is no separation of prothallus, but to promote cross fertilization, the archegonium is usually formed before the antheridium is formed. And this type of prothallus, they are either saprophytic if they are underground, or they are photosynthetic if they are present above ground. But the lower portion of the thallus, whether it is above ground or subterranean, it is always it should be infested with fungus, because without the fungal infection, further growth will not happen. And also, after the archegonium, it has started, fertilization of few archegonium have started, then the growth of the gametophyte is stopped. The male gamete, it is biflagellate or multiflagellate sperm, and it swims towards the archegonium. Here we see in this diagram the mature archegonium, and so the sperm, it swims, and the neck and the ventral region, the cells in the neck and the ventral region, they disintegrate and produces a mucilaginous secretion that is a chemoattractant for the sperm, and sperm being attracted by the mucilage swims to the ventral region and fertilizes with the egg in the archegonium. Now, after that, the zygote and embryo formation, and even sometimes the entire sporophyte, it is formed inside the gametophyte. Here, this one, here in this picture, we see this one is the is a it's a sporophyte that is forming inside the gametophyte, this region. So also this is a picture where inside the megaspore wall, where archegonium is developed, and the sporophyte, it is getting developed inside the megaspore wall, inside the megaspore only. So this is quite common in the pteridophytes. Here we see the precocoious germination in Selaginella, which means that the male that the female gametophyte, that is the megaspore, that do not leave the strobilus while still in the sporophytic plant, the megaspore, it mature and gets fertilized, and the zygote develop into an embryo and another sporophyte. All this thing occur while still inside the strobilus of the sporophytic plant. So this is the sporophytic plant of the new generation, and this entire structure, it is the this entire structure is the strobilus of the parent sporophyte, and the megaspore are present inside this sporophylls. And see the plants, this is the sporophyte of the next generation. This is the sporophyte of the next generation. So this type of germination while still inside the parent plant, it is called precocoious germination, and this is found in most of the Selaginella species except these two species of Selaginella. All these type of precocoious germination are found in all other species of Selaginella. So this precocoious germination in Selaginella, it is very important because short questions usually come. Then we come to the vascular organization, where we see that this is the type of stele, protostele. It is found originally in all the pteridophytes and immature plants of the pteridophytes. But as plant attains maturity, we move on to the siphonostele and the actinostele. When in the protostele, they have xylem here, and they have phloem on the outer side here, and they have an endodermis. When they develop projection, star-like projection, they are called actinostele. This one. And when but the entire stele, there is develop a pith inside it, and the entire stele is surrounded by the endodermis on all sides. It is called the amphiploic siphonostele. And when the pith appears, but the only the outer region of the stele has this endodermis, then it is called the ectoploic siphonostele. That is, only on the upper surface endodermis is present. And later these other type of steles develop from this siphonostele. It later develops into eusiphonostele, and later the eustele, and the amphiploic siphonostele, which has got endodermis on both surface. Here you see the endodermis is present both on the upper as well as the lower surface. They develop into either dictyostele or the atactostele when the due to leaf gap they are dissected and several times they are fragmented by the leaf gap. They develop into dictyostele and atactostele. And when the star shaped actinostele, they get further, they get due to the leaf gap, they are fragmented, they form this plectostele. So this type of stellar organization, they are very important because they are first found in the pteridophytes and very characteristic
the economic
feature of the
importance,
pteridophytes are of immense economic importance. First, I should say they are of immense evolutionary importance, and because they are found even before the dinosaurs, even they are found in fossil records, so of immense evolutionary importance, as they are the precursor to the land plants. And apart from that, they have economic importance, especially as ornamental, because we see in floral decorations many ferns and various type of other pteridophytes are also found, and they form an important part of decoration. Apart from that, there is another their immense ecological importance, that of the indicator plants. For example, they detect for example, equisetum, it detects gold. It accumulates gold in its body, and at the same time, asplenium, it accumulate nickel inside their body without damage. So actinopteris is another fern that is an indicator of cobalt, which means that in it accumulates, its bio accumulates cobalt. So they can be used as an indicator plants, and they planting any of these pteridophytes in an area, we can easily find out whether there is any heavy metal pollution or not, so any metallic pollution or not. And also another fern, that is azolla, which is a water fern. It has got a symbiotic relation with the cyanobacteria anabina, and we know that the cyanobacterias, they are the because they fix nitrogen in their heterocyst, so they can be used as a nitrogen fixer in waterlogged fields and improve the soil fertility. And this as because this is a very organic way of biofertilization, this is also very important. And the pteridophytes have got many medicinal importance because of the secondary metabolites, that is phenolic compounds that it generates. Pteridophyte is of immense medicinal importance also. Also certain fronds of certain species of pteridophyte, they are of importance as a cattle feed. So this was all about the overview of pteridophyte. I know the video was long, but as because it is an overview, so I had to include all the various topics, and I'll be making detailed video of individual pteridophytes, that is in the masters and bachelors syllabus of various universities. And if you want me to make any videos on specific topic in pteridophyte, do write it in the comment section. And if you like my video, do share it with your friends, and please hit the like button if you like it, and also do subscribe, because your subscription, it encourages me. And do write comments on how you like this video, or any other topic you want me to make videos on. Thanks again for watching my video, and please do subscribe.